All right, we're going to get going, and I'm going to speak very loud that we're going to get going so that people out there can hear us that we're going to get going. So welcome to everyone who came to this session, and welcome back from the break, as well as those on Facebook Live. And we will be doing a small opening to this session in a moment, but first I'd like to welcome Jean to speak to a little bit of an intro on her own.
Thank you so much to the Alliance for scheduling this talk and for the wonderful panelists for being here and taking their time to talk about this very interesting and important topic. Before we start really getting into it, I will just share a little bit about the impact of genetic ALS in one family in one generation. This is my mother's generation. They witnessed their grandfather die of ALS in their youth in 1970, and their mother die of ALS in their young adulthood in 1990. Then there was quiet for 27 years, aside from anxiety and fear. So right here, this is my mother, Kathleen. She was diagnosed in 2017 at age 68. She was always open about her fear of developing ALS with her family, with strangers, and of course with her doctors. It still took a year to receive an ALS diagnosis after progressive paralysis started. She pounded the table in the neurologist's office, insisting that she had ALS in January 2017, and the neurologist dismissed her family history, called her 2 drop feet, peripheral neuropathy unexplained, and she would not be diagnosed until she also lost use of her hand and started slurring her words. After my mother's death, my Aunt Joanne was diagnosed at age 58. She had been symptomatic for at least a year before she allowed herself to be seen at the ALS Families Project at Columbia under the auspices of being an at-risk person, but of course, she walked out of that appointment with an ALS diagnosis. She died in 2023. She was the youngest of the generation to make it to adulthood. They had a sister who died tragically in childhood, and my mother was the oldest, and my aunt was the youngest, and there were 5 in between waiting to see who was next. And that was this year. My Uncle William was diagnosed at 74. He had been progressively frail for many months before his diagnosis and was unable to do his post-retirement job as a dog walker. He is still battling ALS right now. My brother just brought to him the ramps my grandfather procured for my grandmother, which my mom kept a hold of for her own eventual use, and now he's getting to use them. So in this one family in one generation, not a year has gone by without a member suffering from ALS for the last 7 years since 2017, though due to the rapidity of the disease progression and the delayed diagnosis, despite the known risk, none of the 3 had an ALS diagnosis at the same time. And this is repeated across many families across the world. Thank you.
Thank you, Jean. So I want to say thank you to the Alliance for this panel and for having the good fortune of being the moderator of this panel. My name is David Taylor. I'm the VP of Research and Strategic Partnerships at ALS Canada, and this panel is about the path to pre-symptomatic ALS MND trials. There are a lot of researchers who believe that understanding the biological disease of ALS upstream of symptom onset is going to be our best opportunity and chance to be able to make a headway towards substantial treatments for the disease and maybe someday a cure for the disease. But it is really complex. And so when we think about how that's going to happen and how do we coordinate that around the world, we should first take a step back and take that first step towards explaining it to an audience at the Alliance about what some of those challenges and hurdles are. And when you think about what does that mean, the biological disease upstream of symptom onset, many people think of the disease as starting at symptom onset. Well, it was actually visionary science and work done by one of our panel members more than 15 years ago to start working with individuals who are at high genetic risk to understand the disease that is happening upstream of that symptom onset that allowed us to have a panel like this today in 2024. And it is the need to be able to step into that today that will eventually allow us to understand that disease upstream of symptom onset and to be able to get to those treatments faster in the future. And so there is no better panel that you could find in the world for this, representing academia, the ALS community, and industry to be able to speak to this important topic. And it's something that I suspect will be coming up at Alliance meetings year after year, so we can look at this as some of the first steps into that. We're going to hear probably some of the challenges that are with exploring asymptomatic, pre-symptomatic clinical trials for ALS MND, and we're going to hear some of the boundaries that should be pushed, and I think that's what makes for such a great panel. So to introduce our panel on the academic side, first we have Professor of Neurology and of Public Health Sciences at University of Miami Miller School of Medicine, Dr. Michael Benatar. Dr. Benatar is that individual I talked about who had that visionary idea more than 15 years ago that has really paved the way for these conversations we're having today. Next to Dr. Benatar, needs no introduction, Gene Swidler is the Executive Director of Genetic ALS and FTD and the Legacy. Only in the past few years has become an absolute tour de force when it comes to advocating for people at high genetic risk. And her acumen at being able to understand the literature, to meet people where they're at, to show an incredible ability to integrate what she wants to do to push those boundaries, but at the right way, that has put her at this table today, and we're so grateful that she's here. And next to Jean is Stephanie Fradette. Stephanie is the Vice President, Head of Neuromuscular Clinical Development at Biogen, and she'll hate that I'm going to say if you all are very aware of tofersen Qalsody being the real universally accepted way that we can now understand that ALS is a treatable disease, there is likely no one in this world who has put in more effort to get that to people than Stephanie Fradette, and we are very grateful, and she's probably one of the most respected people in industry in ALS as well. So this is a really awesome panel. So we're going to start, and we're going to give each of the individuals, the panelists, a couple of minutes, probably about 5 minutes each, to sort of talk about what would be that first bit of conversation you want to talk about. What are the challenges and hurdles that we face in terms of approaching asymptomatic, pre-symptomatic clinical trials, some of the things that are already going on we'll hear about, and we'll go from there. And then please do think about questions, because we're going to open Q&A for this discussion after we have these opening statements. So please, passing it to you, Dr. Benatar.
Great, thank you, David. Is the audio okay? Great. So, I think this panel is entitled The Path to Pre-symptomatic ALS MND Trials. And in reflecting on that, I think it's helpful to look forward, but it's also helpful to look past. And maybe to underscore that for unaffected carriers of SOD1 mutations, that future is now. That trial is now ongoing. But it's helpful to understand how it is that we got there. This trial, I think, has been possible because of some of the foundational work that we had done through the pre-FAL study, trying to understand the pre-symptomatic course of disease, identify biomarkers that might tell us when disease is coming, and then through a fantastic partnership with Biogen, as tofersen emerged over the horizon as a safe therapy, as an effective therapy for people with manifest SOD1 disease, we were ready to say, let's put these knowledge pieces together and design a pre-symptomatic trial that might aim to prevent disease. And the key thing is that when we started pre-FALs back in 2007, we had this idea of prevention in mind, but we knew that there was much more that we didn't know than we did know that would be essential for us to know if we were ever going to do this sort of study. And the key was to say, let's not wait until we have a potential therapeutic that's worthy to study in mind before we lay that foundation. And so laying that foundation meant that when the drug candidate emerged, we were ready to go. And as ready as I think we were for this subset of the SOD1 population, I don't think we're quite ready for other genetic forms of disease, and certainly not for non-genetic forms of disease. But we need to be learning from that experience of laying that foundation now so that when those therapeutic candidates emerge, we're ready to go. So we can come back and talk more about that, but maybe just to say for the most common genetic cause of ALS, the C9orf72 repeat expansion, as this group well knows, people are at risk not just for ALS, but also for frontotemporal dementia, and that's true of other genetic causes of disease. So when we're thinking about pre-symptomatic trials, we have to think about what are we preventing, and it's not just ALS. We're also trying to prevent FTD, and really any phenotypic manifestation of what that genetic risk in this case, or risk, might put somebody at risk for. And to your point, David, that's why we have to understand the biology and develop treatments that target that biology. And to the extent that we do that, we'll be working towards preventing not a single phenotype, but any phenotype associated with that underlying cause. But I do want to underscore as well, so I realize I was supposed to ask you to put up a slide. I forgot. So there's a slide if anybody wants to follow along. So we'll bring that up. But I do want to say that I think, and I'm hoping this panel will go beyond genetics as well. I want us to open up our minds to the possibility that we should be thinking towards the future of pre-symptomatic studies and prevention trials in a non-genetic population or a non-genetically at-risk population. And the way to do that is that we have to identify the risk factors for the non-genetic forms of disease. The reason we started with the genetics is because that was the tractable place where we could start, where we could get a handle on studying people at risk for disease. And what we surmised, and I think we now know, is that lessons that we learn will have broader relevance. It won't be the same solutions, the same tools for every case, so for every genetic form or non-genetic form, but the principles will apply, and they're things that we can learn. And maybe the last thing to say is that success now and success going forward, I think, is critically dependent on partnerships. It requires partnerships between the ALS community, the FTD community, and other disease communities. It requires partnerships between academia and industry. It requires partnerships that engage stakeholders across the spectrum, the carrier community, the regulatory agencies, academia, industry, all of those. And so I think as we're looking towards that future, I just want to underscore the critical importance of those partnerships. And I think we'll come back and talk about many of these things, but I'll stop there.
And Jean?
Thank you so much, Dr. Benatar, for laying all that out. I'm so thrilled that there's brilliant, compassionate, dedicated folks like yourself and Dr. Fradette working on this, so thank you again. I think my brief comments here are going to definitely be more of a general position on these things, so forgive me. So in my opening, I shared the nature of the existential risk in my family, and that exists in so many other families, and that's an important thing that is not great. But I will stress the need that not just for our families, but for the entire ALS community, testing drugs earlier in the disease process is important for us to try to make progress in this terrible disease. Because we have had failure after failure after failure after, supposed success, which was really a failure in drug development for people impacted by TDP-43 ALS, which is 97% of ALS, which is all of sporadic and most of genetic, including C9. And we really need to cure this disease and make huge impacts on disease progression along the way. Testing drugs earlier in the disease process, which is currently only practical in those with a high genetic risk, would allow for more definitive answers if any possible therapeutic approach works better or only earlier. And while we may be tempted to think only of possible pre-symptomatic trialing of drugs as standalone trials, like the ATLAS trial for SOD1 that's happening right now, the most practical path forward for beginning to learn about the impact of possible approaches earlier is using smaller cohorts of genetic carriers with some confirmation of biological disease in otherwise larger studies of drugs in symptomatic ALS patients. This is a tried and true approach in rare drug development, and cohorts and trials that do not contribute to the pivotal clinical efficacy question can help with questions of accelerated approval or guide follow-up trials. We only need to look at the Valor study for this example, where the pivotal question was the impact of the drug on a fast-progressing subset of the full trial cohort. While that pivotal clinical efficacy question did not meet its threshold, the trial received accelerated approval based on the NFL response in the full cohort. So the patients in the study who were not part of the main efficacy question contributed to the accelerated approval. So let's follow that example in all late-stage ALS trials. And then a few loose notes. One is about the FDA. The FDA has made clear that they support experimenting in earlier cohorts. They've made lots of statements and many meetings about this. And then also, they have approved trials with pre-symptomatic cohorts with biological disease activity in progranulin FTD and in a small phase 2 with C9orf72 carriers for baricitinib at NGH. But I realize the willingness of the regulator is not the only issue here. And secondly, the group that we are considering for this are not healthy people. They might be fully functional people, but we should only be considering trials in people with signs of biological disease as an inclusion criteria for it. It has been established for many decades that motor neurons are extensively damaged before any functional impairment is reached, and this may be the reason drug development has been so underwhelming in ALS over the years. Just briefly to speak to safety, certainly for novel medications with uncertain side effects, establishing some initial safety in symptomatic patients seems reasonable. So really just looking at late-stage, larger trials with smaller genetic cohorts would be what might make sense. And finally, for biological signs of a disease, it may be true that much more is needed to have full confidence in what the biological trajectories mean. However, if uniform biological response is seen at the levels observed in Valor in a biologically active pre-symptomatic cohort, that would be compelling data that could be created to support a full package for the FDA combined with positive outcomes in a fully symptomatic cohort, or clear need for a pre-symptomatic confirmatory trial if there is a separation in the response between those earlier and later in the disease. Thank you so much.
Thank you, Jean. Break the microphone before I start. I asked Dave if I could go last because I see the world very similarly to Dr. Benatar and Jean, so I didn't want to I won't be redundant, but just a couple of thoughts. I think, the ATLAS study, aside from my children, is probably the thing that keeps me up the most nights. It's a tremendous honor to be part of running this study, but it's also a tremendous responsibility. And the learnings that we are accumulating in real time are something that we're continuously trying to share with the world, because while this study, and we've alluded to this, but is in a specific subset of the SOD1 population, faster progressing variant carriers of SOD1 variants, and while it's specific to that population, the learnings from the study, of course, are sort of paving the way for how to do this going forward. But as Dr. Benatar alluded to, we can only do that if we understand what are those factors that are moving earlier in the disease process in other subsets of the population. And we were only, as both Dave and Dr. Benatar said, we were only able to run the ATLAS study because of our understanding of what was occurring in these at-risk, quickly progressing SOD1 variant carriers in advance. It's the only reason we could get buy-in from regulators to run the ATLAS study. And so we have a lot more work to do to understand these factors and like I said, what is moving in advance of clinically manifest disease in the broader ALS population. So everything we can do to generate and build that understanding. I know there's a lot of ongoing work across the globe to characterize the natural history in these at-risk carriers, and that work is absolutely imperative. I think the ATLAS study will teach us a lot about tofersen, but what excites me most is what it will teach us more broadly about the disease. We talk about sort of the fact that we will have CSF samples from these at-risk carriers prior to elevations in neurofilament. So when their disease process has not yet started, what will we learn there? What will we learn on comprehensive EMGs in those individuals? How will that differ at the time that neurofilament levels have gone up? And so some of these factors, if we're looking at levels of SOD1 protein and things of that nature, may be specific to the subset of the population that we're evaluating in ATLAS, but many of these factors will not be. One of the perhaps most important things that we're experiencing and learning and need to do more of there's a recent publication that both Jean and Dr. Benatar were on about monitoring of at-risk carriers in the real-world setting. Because one of those things that keeps me up at night is as we have individuals that are not eligible for ATLAS or that are part of ATLAS but don't ultimately make their way into the interventional study period, then what for those individuals? So the more that we can have global partnerships to ensure that those individuals are getting the monitoring that they want and need, the better. And if we can learn from that experience, then I think we all win. I think that one other thing that I'll mention is, and this has been alluded to by all of the panelists, we run the risk that we are intervening too late. We recently had an ATXN2 readout, and there are a number of different reasons why that ASO may have failed. One of the hypotheses is that we intervened too late. The preclinical data that we have on ATXN2 are primarily in the pre-symptomatic setting. And so if we could intervene earlier with some of these targets, maybe not pre-symptomatically, but even at the time of early symptom onset, are we going to be able to more robustly test these scientific hypotheses and remove one of the most important confounders, because we know time matters in these individuals. So while we're focused on pre-symptomatic intervention, we're also needing to do more to understand those early clinical signs and symptoms, and this has been a focus of Dr. Benatar as well. So I think the more we can define that early symptomatic period, the more we can enrich clinical trials, particularly if we have, as Jean alluded to, objective biomarkers that aren't making us dependent on progression clinically. We can run different clinical trials and be more inclusive in those clinical trials for the broader population as well. So just a couple of thoughts to touch on.
Please, if you'd like to, before we get into any questions, sure.
Yeah, I just wanted to add a comment and a thought. I think, when we're trying to identify an effective therapeutic, we have to have a signal that can tell us that that therapeutic is meaningfully effective. And the catch-22 that we're in is that we can measure that therapeutic signal more readily in the affected population, but it's also the population in which the disease may be too advanced for the therapeutic to work. And when we're in the pre-symptomatic population, until we have those good biomarkers that are something we can measure biologically, we don't have anything that we can measure, and so we can't judge and evaluate whether a therapeutic is having an effect. And so maybe one of the most essential keys to get us to a future of more pre-symptomatic trials is development of quantifiable biomarkers in the pre-symptomatic state that we can use to tell us, is the drug having an effect? And whether that's, for example, including carriers, unaffected carriers in a clinical trial primarily focused on the affected population, we still need to be able to measure that effect, and the biomarker will be key.
And so I just wanted to also level set for everyone in the audience. When we think about the trial that's been talked about here, the landmark ongoing ATLAS trial that's happening, at a very basic level, when we're thinking about this, from the work that Dr. Benatar has done, it has shown that people with a specific subset of variants with SOD1 variants will typically convert to having symptoms in approximately 1 year. And I know that's a very crude overlay of saying it, but the idea here is that if they are able to be treated with a therapy that we know is effective that far in advance, when there's a signal of a spike in neurofilament, which is representative of neuronal damage, there's the opportunity or the desire that this may actually have people going well past the time when they typically would have got the disease. And of course, we all hope that this may show that people who have a history of fast progressing disease may not get the disease. And that is what we're talking about here, which is incredible. And something 5 years ago, just you would have conceived of it, but a lot of us would never have done that. So I think that for an inspirational piece, that's really why we're up here, is to think about how we can do that for everybody. And as you said, even the non-genetic or at least non-obvious genetic individuals with the disease. Before we get into the questions for the audience, and please do think about your questions, I just have one additional question here for the panel, and that is, okay, so we know that for other individuals at high genetic risk, or even those who are more complex with terms or with regard to how their disease starts, what would you say the research community, given that there's a lot of organizations supporting research out here, should be doing globally, collaboratively, to try to support doing this as quickly as possible and understanding the complexities of the other forms that we can try to approach in the same way. And then, of course, I would also like to add to that, if Jean could talk about, as we get to those things that we should be doing, what should we be mindful of for the community who we might be having volunteer for a lot of those studies to effectively do it in the best way. So perhaps, Michael, you had said it's complicated. Perhaps you could help us understand what we could be doing.
Yeah. Maybe I can sort of use an example to try to illustrate a broader point. I think we need, if we're going to understand, if we're going to have a future of pre-symptomatic trials, let's at least say for now in other genetic populations, we need to study the carrier community at scale. But it's not just at scale, it's also with the relevant depth and care of clinical phenotyping and biological phenotyping is the wrong word, but biological understanding. We want speed and we want scale, but not at the cost of quality, because I worry that if we run too quickly, we run the risk of investing an enormous amount of time, energy, resources, and then we turn around and we say we don't have the answers that we need. So, for example, we've been able to identify prodromal stage of ALS that we've called mild motor impairment, which to this community will come as no surprise that developing overt ALS is not like flicking a light switch. It's not like it's not there and then it's there, right? It's more like a dimmer switch is the analogy I give people. Biology is a continuum. The disease is gradually emerging, and then it sort of appears above the clinical horizon and becomes clinically apparent. We didn't know that when we started, and for years I was scratching my head saying, I no longer think I know what ALS is, because I'm seeing all of these, in scare quotes, abnormalities, but I don't know what they are. This is not ALS. And it was only through the careful approach to phenotyping that we could identify a prodromal state. Now, to the extent that we're right about that, maybe that then becomes studyable in other genetic populations and even non-genetic populations. I'm thinking here by analogy to mild cognitive impairment in the Alzheimer's world, and our clinical trials in the Alzheimer's world have moved into that earlier prodromal stage of disease. And so we didn't set about to discover a prodromal stage, but we set ourselves up to be able to identify early markers through a careful approach to phenotyping, and knowing that disease could emerge in an arm, in a leg, the right, the left, the bulbar, the respiratory apparatus. And so we knew we had to broadly phenotype to understand that. The same is true if we're looking biologically. We don't know where that signal is going to come. Is it going to be blood, plasma, serum, RNA, CSF, DNA, urine? So we have to think broadly in terms of setting ourselves up for that success. And so I guess what I'm trying to say is we want to do this at scale and we want to do it quickly, but I want to make sure that we collectively think about what we need to set ourselves up for success.
And I'll just double click on 2 points that Dr. Benatar mentioned. One is sort of the scale bit, which I think what you have done at the University of Miami is unbelievable. The ability of other individual institutions to do the same thing is inevitably going to be more limited for the broader population. So the more that groups are coming together and joining data sets and testing samples with a single assay versus everyone having their own assays that they're running where we can't actually integrate data across these different studies. So the work that Precision and AMP-ALS are doing and many others like it, I think is imperative to get to scale. And then to Dr. Benatar's point about quality is also frequency. And that's something that we're learning in the context of the ATLAS study is the importance, although it can be burdensome, of having frequent data collection points to really understand and characterize this early part of the disease process. And so we have to figure out what that ideal interval is, but it is something that we have to consider more broadly.
So just to jump back in, thank you guys so much for your well-reasoned and argued thoughts. I just wanted to stress that right now there's drugs being trialed in ALS where the outcome in the fully symptomatic cohorts are leading the field to exclude hypotheses of what the disease is. And without testing earlier, being as a hedge to show that that's not what the issue was. And there are, something like a few thousand C9 carriers developing ALS or FTD every year in the US, more globally. And there is published evidence from the FTD cohorts that there's a 3-year there's a significant separation from controls for C9 carriers who are going to develop disease 3 years previously. And I realize that's not really ready for prime time for like a full accelerated approval, most likely for C9. But if there's people in that position with an elevated NFL that is significantly different than controls and age-matched controls and all of this, and there's drugs that need to be trialed, and we can find 20 or 30 or 50 of them with proper disclosure of their NFL results, just to see if there's some huge difference in the biological response in the symptomatic versus the earlier people, that would tell us something. If there's 30 people with a uniform response, that might not be statistically significant to say that it definitely works, but it would be a clear signal that there's something there that needs to be pursued, whereas right now, none of that's happening. So that's kind of where I would just frame it.
Yeah, perhaps I can just say, I partially agree with you. The devil is in the details. So the all-FTD data is very interesting. But what they show is that at a group level, on average, neurofilament levels are higher in the carrier population than a non-carrier population. That's quite different from being able to say, this individual, their neurofilament is, quote unquote, elevated. So showing a group difference is not the same as showing that an individual is elevated. And so if one wants to use that as a response marker to know whether you've impacted that, you have to know that the people you're enrolling, that they individually are at risk of whatever that outcome is, or that there's a measurable substrate to understand. So I agree with you, but I also worry that we might run before we walk with some of those data because of the group versus the individual level interpretation. And I think that also depends on how we then use that information. If it's to sort of inform a broad understanding of biology, or if it's to inform selection of individuals to be enrolled in a trial, those are sort of different utilizations of that information. And I think it can lead us in different ways, in different directions.
Thank you so much. I just want to affirm I completely rely on you and your colleagues to put the fine point on the details here, but there's just a lot coming out about imaging markers or connectivity markers and blood markers and stuff. There's things that could be looked at, and if there is no response on those markers, I think that's probably a sign that it's not a home run. That's not to say that it wouldn't be something at the level of a riluzole or something, but for a huge effect, we would expect to see a very clear and dramatic biological effect. So just some thoughts.
Yeah, and I think, sorry, Dave,
No,
I know you want to move on. I agree with both of you. I think that the bar for incorporating a biomarker or imaging marker for determination of eligibility is much higher than I realized when we were designing the ATLAS study. But I think that's why understanding sort of the natural history is so important. And I don't think we're that far away from trying to enrich a cohort. To state the obvious, we need a drug that then impacts that marker, which is the other part of the equation that I think we're all working towards, is having a drug that can meaningfully affect objective markers of the disease process such that we can make an informed decision. But it's something that we think about all the time. And to be completely frank and transparent, it's something that we contemplated for, had we moved forward on the Ataxin-2 program, for example, is there a way of looking at a C9 cohort earlier in the disease process in more of an exploratory manner as part of that pivotal study? So it is something that we're actively contemplating, and I think we have to zero in on how to meet that threshold that inevitably regulators will require for identifying the population.
Just point of order, there was, this is me merely as an observer, there was this baricitinib study last year at MGH where they had an inclusion criteria of some inflammatory markers for 5 pre-symptomatic C9 carriers. I don't understand. I have never seen anything clearly published about inflammatory markers for pre-symptomatic conversion, but the FDA let them do it. So just raising that up.
So I want to, if anyone has a question, please raise their hand. But I want to also, what you're seeing here is incredible dialogue over a very complex situation. And I think that's what's amazing here. And it might be unlike other situations where Michael and Steph and Jean talk about this a lot with each other. And what you're seeing is a snapshot of the conversations that they're constantly having between how do we do this appropriately? How do we meet the urgency that we need to be able to push the boundaries, like Jean's talking about? And how do we integrate with industry to be able to make this actionable? So, I mean, I find it fascinating. We couldn't even get to half of the questions that I had written down, but that shows you why what I was saying is we're just in the very, very opening steps of starting to have these conversations, and having them ongoing is incredibly exciting and hopeful. But there's a lot to be done to figure out what that happy medium is between doing it right, but also the urgency that is needed and the care that's needed in doing it. Because Jean, I know you and I had conversations in the past where you explained to me, and that, you know, these studies also need to have give back for the communities who are going to be participating in them, and that comes back to your regular monitoring and pushing boundaries and thinking outside the box and all of that. And I wanted to make sure that that gets highlighted too, because it's absolutely critical that we're not talking about people at high genetic risk as just responsible for us to figure out the rest of the disease. And you've definitely learned me much on that over the years, and I appreciate it very much. Does anyone have any questions in the audience? Or we'll just all keep talking up here for...
We really like each other, by the way, just in case that wasn't obvious. We're learning from each other as we're talking.
This is healthy academic dialogue, which is incredible. Is there any questions?
I have a question here.
Yes.
Yeah, my name is Debbie Lauer, and Dr. Benatar, I first got to listen to you in 2013 at ALSA when you did a familial ALS presentation. And I am so excited to see the progress, you know, over these last 11 years. And when I see, you know, the idea of doing a trial for C9, that's what my family has. And I think about what Allison just talked about, that you can go out to rural, you know, in my case, America, and my family, it puts their head in the sand when it comes to, you know, trials and studies and that sort of thing. I have over 120 living descendants from my grandparents. So it took 5 of 8 in my mother's generation. We've already lost 2 in my generation. Now number 3 was just diagnosed, and number 4 will be later this month. So do you have any advice for someone like me who's tried desperately to get my family engaged? I've met with them 4 times over the last decade to teach them what to look for and all about studies and trials, and I'm not getting through. So I would love to know if you have any advice for how to break down that barrier that Allison talked to us about. Thank you.
So thanks for the question. I'm happy to start, but my perspective is only a very small part of this. So I guess my perspective from Pre-Fals is that when we started, we wanted to be as broadly inclusive as we could and enable people to engage in the way that they were comfortable. So what that meant is we offered people could join the study if they had undergone testing and knew they carried a gene. We offered people who wanted to participate and learn if they go through pre-symptomatic testing and learn that. We also said, if you're just from a family and you want to participate, but you don't want to go through genetic testing, that's okay. We'll do that on the side, and only our stats people will know about that. So we wanted to be broadly inclusive. So I guess that's the one message, is as we're designing studies, we should try to embrace as broad a segment of the community as possible. What we've also seen when we transitioned from not only running Pre-Fals, but then to enrolling for Atlas, is now the motivation changed for many. And so now people said it's not just an observational study, it's also an interventional study, and that brought in an additional segment of that carrier community. And it took some of the people who had decided they wanted what we call non-disclosure to participate without learning genetic results to say, now's the time for me to learn. And so I don't know that I have a clear answer or one size fits all, but maybe to the extent that it's true and it should be, to say that the research community is looking to engage you on the terms that you want to be engaged on, and it's our responsibility as a research community to rise up to meet that challenge and to find ways for people who want to participate at whatever level, whether it's a remote visit once a year, whether it's an in-person visit every 3 months, whatever it is, we can tailor that to enable as broad a segment to participate. But I'm sure others will have other perspectives.
Well, Debbie's a friend, so we talk all the time. So I'll let Steph go.
I think your perspective is spot on. I mean, I don't have anything much to add. I think that different motivations will be relevant for different people. I think if there's anything that we can do to help educate and make people aware of the fact that we would not be running this ATLAS trial without the contributions of the pre-FALS participants. It would not be happening. Whatever we can do to shout from the rooftops to help, others understand that, that reliance, I think we'd be very happy to do that, but it goes without saying that we wouldn't be where we are. We wouldn't be having this conversation or running that trial without those individuals and their contributions. So.
And maybe just to add one other thought is to encourage your family members and that broader community to reach out to researchers and learn about whether there's an opportunity to engage on their terms. Not to prejudge it and say that it's going to require X or Y or Z. And certainly if they reach out to us, if we don't have an opportunity for them to engage, we will try to help connect them with other groups where there might be opportunities. So I think to the extent that you can help get them to over that initial hump and just some contact and discussion, then we can try to meet them where they want to be.
And more for the audience, Debbie's heard this, but thank you, Dr. Benatar, for leading the project with Dr. Hyman Patterson about the guidance that just got released for the very first time, which tells people that there's some little things that they should consider doing for their own personal health while they're at risk, which never existed before. So that's new information that we have to make sure that any family is aware is out there if they want to learn about it.
And maybe to say that's another opportunity. If the carrier community starts coming to have their care needs met, maybe that creates an opportunity to learn about research opportunities. We see this in the clinically affected population. They come to multidisciplinary clinics and they learn about ongoing trials and research opportunities. To the extent that we start to build a network of clinics and clinical service for the carrier community, maybe that creates another opening or opportunity.
Great. We have time for one last question.
Okay, I'm going to rock your world for a second. And I'm friends with Debbie too, so this has nothing to do with it. But this is exactly the bias that we uncovered on our trip, is we assumed everybody wanted to know that information. And from an ethical perspective, once you take away their piece of not knowing it, they can never get that back. And so where do you cross that line to say, these people don't want to live in fear and they don't want to know, and that's going to be okay?
Allison, just to be clear, if they are putting their fingers in their ears, they're living in fear if they're telling you that. And we're not talking about Dr. Benatar or Steph, Dr. Fradette going out to these people and telling them that there's new guidance. We're talking about families, educating families that there's new information. And people in their own families can judge what is best appropriate to share with their family members.
Yeah.
It was an exact family where the kids did not want to know, and they'd lost their mom, their uncle. They knew that there was a genetic component. They didn't want to know about genetics.
But Allison, that was their family's business. I understand that you...
Yes, that's what I'm saying too, is that we need to ethically let people live in peace if they value that too.
Yeah, so Alison, one thought, one of the things we learned along the way when we first started Pre-FALS, and when we were doing this back in 2007, the idea of doing telephone genetic counseling was almost unheard of. I had to strong arm my genetic counselor into doing this with me. But one of the things, we started from the Huntington's model, where we said we have to give people pre-test counseling and post-test counseling. Pre-test deciding whether you're ready to receive genetic results, then you have a test and you come back and you get post-test. And one of the things we learned is that we needed to introduce a third counseling session, what we call pre-decision counseling. So somebody is just beginning to toy with the idea. They don't know whether they want to engage in this. Let's have a discussion about what it means, what that information would mean to you, to your future. And then if they then ponder that and say, I would now like to go forward, we would then still engage in a traditional pre-test counseling. And so I think that was one of the things we learned, is that we needed to give people sort of an extra step along that journey and an opportunity to change their minds at any point, and to underscore that you can change your mind at any point until you actually get the result. And once you have the result, there's no going back, right? But you can be sitting down for your post-test counseling and say, I don't want to know, and that's okay.
And we're going to have to cut it there, but thank you very much. As you can see, there's so much to talk about, and I hope the Alliance continues to make space for this very, very important topic. And thank you so much to the panel. Please join me in saying thank you to them. And thank you to everyone on Facebook Live. We'll be cutting off now, and I'll pass it back to Richard.